With the high volume of samples that are received by clinical laboratories each year, the chance of isolating a biothreat agent is slim. As a result, characteristics of potential biothreat agents often get forgotten or overlooked. However, isolation of a select agent can have many consequences in the laboratory, which we will review later. In this webinar, we are going to review some of the general characteristics of biothreat agents that a clinical laboratory in New York State might encounter. We'll begin our review with Bacillus anthracis. Bacillus anthracis is a zoonotic organism that is most commonly associated with animals such as cattle, sheep, and goats that come into contact with soil contaminated with Bacillus anthracis spores. Human anthrax infection is a result of direct contact with the spores and can cause 4 different types of illness based on the route of infection: inhalation, cutaneous, GI, and injection. Anthrax is not contagious from person to person. Inhalation anthrax, or wool-sorter's disease, is the most fatal form of illness and most commonly affects individuals who are veterinarians, tanners, or other professions that involve work with animals. Cutaneous anthrax is a result of spores entering the body through a break in the skin and is characterized by the classic black eschars, or painless necrotic ulcers, that develop as a result of infection. GI anthrax occurs as a result of ingestion of the spores, and symptoms of infection are similar to those of other significant GI illnesses. Depending on the route of infection, Bacillus anthracis can be isolated from sterile sites such as blood and CSF, as well as from wounds, sputum, and stool. In New York, we have been fortunate to not have many cases of anthrax recently. As some of our more experienced laboratorians may remember, back in 2001, we had several cases of anthrax due to intentional contamination through the U.S. postal system. Since then, there have been several cases of anthrax, mainly cutaneous and some inhalation, in New York City and surrounding states that were associated with individuals making drums in small spaces without appropriate personal protection. It's important to remember because we have such a diverse population, it wouldn't be surprising to see additional cases in people who inappropriately handle hides or import meat. So it's important to be mindful of the characteristics of Bacillus anthracis. Bacillus anthracis grows much more quickly than the other biothreat agents that could potentially be isolated in the clinical microbiology setting. Growth can be observed on blood agar after 18 to 24 hours incubation and 5% CO2, and colonies are typically 2 to 5 millimeters at this point. Bacillus anthracis is an aerobic, non-hemolytic organism, and the colonies have a very flat or slightly convex gray-white appearance with irregular edges that resemble ground glass. Colonies can appear comma-shaped or be described as medusa heads. Bacillus anthracis is also very tenacious, meaning the colonies keep their upright form after being manipulated by a loop or needle. Examples of this can be seen in the bottom picture. On the gram stain, Bacillus anthracis will be a large GPB that can show subterminal and/or central spores. Organisms stained directly from a colony typically have a singles and/or pairs arrangements. However, they tend to form long chains when grown in liquid culture, such as blood culture, thio, or other enrichment broths. Motility and catalase can be helpful in identifying the organism, with Bacillus anthracis being non-motile and catalase positive. When a gram stain is performed from a culture isolate, usually no capsules are present. However, spores may be seen in older cultures. Some organisms that can commonly be misidentified as Bacillus anthracis, either by conventional or automated methods, are other Bacillus species or Bacillus cereus group. It is important to note that some automated ID systems may not alert beyond the group identification. However, it's relatively simple to differentiate Bacillus anthracis from other Bacillus species based on biochemicals, colony morphology, and appearance. B. cereus and B. thuringiensis are still flat and ground glass-like, but the colonies are more yellowy and are also beta-hemolytic. B. megatherium and B. subtilis can be differentiated based on their colony morphology as well as their positive motility. In the pictures, you can see the beta hemolysis of B. cereus and B. thuringiensis, as well as the very mucoid appearance of B. megatherium. At Wadsworth Center, we routinely get roughly 200 samples for rule-out Bacillus anthracis, which is good because it shows that clinical laboratorians are following the appropriate algorithms to refer suspicious isolates. We can also perform speciation for Bacillus species when clinically relevant. In the fall of 2016, the CDC added a strain of B. cereus to the Tier 1 Select Agents list, B. cereus biothreat. This organism has been found in elephants and goats in certain African countries. It possesses the virulence factors that Bacillus anthracis has, and it has been shown to cause anthrax-like illness in gorillas and chimpanzees. It is catalase positive and non-hemolytic like Bacillus anthracis. B. cereus biothreat strains are usually motile, including those recovered from gorillas, chimpanzees, and elephants. But B. cereus biothreat goat strains from the Democratic Republic of the Congo are non-motile. This organism has been infrequently isolated, but it is possible. In 2006, there were cases in Texas where the organism was isolated from metal workers, which did result in fatalities of both workers. Next, we will discuss Francisella tularensis. Francisella tularensis is a causative agent of tularemia, also known as rabbit fever. It is a zoonotic pathogen with hosts that can include rodents, mammals, birds, and amphibians. Francisella tularensis can cause different types of illness in humans based on the route of infection. Infection can occur as a result of being bitten by an infected host, contact with an infected animal, ingestion of contaminated food or water, or inhalation of infectious aerosols. Listed are different forms of illness along with some of the most common symptoms associated with each. Pneumonic tularemia is the most serious form. Not only does this form result from inhalation of the organism in some form, but it can also develop if other forms of tularemia are left untreated. Francisella tularensis can be isolated from clinical specimens including tissues and aspirates, blood, serum, bone marrow, and respiratory secretions. In New York, we have not seen cases of tularemia in recent years. However, there have been recent cases in Massachusetts as well as other states like Colorado. The laboratorians should remain vigilant in the recognition of organism characteristics. Francisella tularensis is a fastidious, classically slow-growing organism. The first signs of growth are usually only observed after 48 to 72 hours of incubation at 35 degrees Celsius and 5% CO2. While poor or pinpoint growth may be observed on blood agar during this time frame, the organism grows best on chocolate agar or another form of cysteine supplemented media, such as modified Bayer Martin, buffered charcoal yeast extract, or a style glycolate broth. On both blood and chocolate agar, colonies are opaque and gray-white with a smooth and shiny surface. Here you can see the slow-growing nature of Francisella tularensis on blood agar, which is shown on the top row, and chocolate agar shown on the bottom row. After approximately 48 hours, growth only reaches approximately 1 to 2 millimeters. Gram stain of Francisella tularensis will show tiny, pleomorphic, gram-negative cracobacilli, usually arranged as single cells. Typically, you may see poor counterstaining with safranin. However, basic fuchsin counterstain may increase the resolution. It's catalase negative or weakly positive, oxidase negative, beta-lactamase positive, and it's also negative in the satellite test. There is potential for Francisella tularensis to be misidentified as other organisms that are more routinely isolated in the clinical laboratory. These are some examples of those organisms, along with biochemicals and tests that can be performed to help either rule in or rule out Francisella tularensis. Next up is Yersinia pestis. Yersinia pestis is the causative agent of human plague. It's a zoonotic organism endemic to the western United States, most commonly in New Mexico, Colorado, Arizona, and California. Yersinia pestis is commonly associated with rodents, such as rats and mice, and it is spread to humans via being bitten by an infected flea that also came into contact with an infected rodent. However, it can also infect humans if the organism comes into contact with a wound or break in the skin. Plague can manifest in 3 different forms of infection: bubonic, septicemic, and pneumonic. Bubonic plague commonly causes lymphadenitis or buboes, and it is the inguinal lymph nodes that are affected in most cases. Septicemic and pneumonic forms can also occur, but in most cases they are usually secondary to the bubonic form. Pneumonic is the most fatal form of infection, and this form is contagious. We have not seen a case of plague in over 10 years, but with the frequent travel of New York State residents to the Four Corners region, it is important to recognize the characteristics of this organism. Yersinia pestis is a facultative anaerobe normally isolated from sterile sites such as blood, CSF, and other body fluids. It will grow in 5% CO2, but it prefers an ambient atmosphere at room temperature. It will also grow at routine 35 to 37 degrees incubation temperature, but growth rates will be significantly slower than other Enterobacteriaceae at this temperature. Growth can first be observed on blood agar after 24 hours of incubation as pinpoint to small, gray-white, translucent colonies with little or no hemolysis. Small pinpoint non-lactose fermenting colonies may be visible on McConkey after 24 hours as well, but may take longer than that to show up. At 48 hours, gray-white, slightly yellow, opaque colonies can be seen on blood agar. The colonies are pretty sticky or stringy, and they usually have a characteristic fried egg or hammered copper appearance. Here you can see the fried egg appearance with little to no hemolysis. After approximately 48 hours, the colonies tend to be roughly 1 to 2 millimeters in diameter. Yersinia pestis on the gram stain will show plump gram-negative rods arranged mostly as single cells or pairs with bipolar staining, although that characteristic is not unique to this organism in comparison to the other Enterobacteriaceae. The safety pin appearance that is not seen on gram stains may be exhibited by Giemsa stain or Wright stain. However, when the organism is grown in liquid media, such as broth or blood culture, it can form chains, which is not usually typical of the other gram-negative organisms in this family. Biochemically, Yersinia pestis is catalase positive, indole and oxidase negative, and urease negative at 35 degrees Celsius. Some possible misidentifications for Yersinia pestis can include Acinetobacter, other Yersinias, including Enterocolitica and pseudotuberculosis, and non-lactose fermenting E. coli. Acinetobacter species can usually be differentiated based on their different colony morphology. Yersinia enterocolitica and pseudotuberculosis can be differentiated based on their positive urease reaction, and non-lactose fermenting E. coli can be distinguished by its faster growth rate and its positive indole reaction. Yersinia pestis may not be identified in common automated ID systems, including MALDI-TOF, and additional possible misidentifications may include Shigella species, non-sulfur producing Salmonella species, and some Pseudomonas species. Next, we will focus on 2 Burkholderia species, Burkholderia mallei and Pseudomallei. The 2 Burkholderia species that fall into the biothreat agent category are Burkholderia mallei and Burkholderia pseudomallei. B. mallei is the causative agent of glanders disease. While human infections of B. mallei are rare as the organism primarily affects horses, infection can be fatal without treatment. An interesting note about B. mallei is that there is documentation of its use as a biowarfare agent in World War I. B. pseudomallei is most commonly associated with soil and stagnant water in the areas of Thailand, Vietnam, Northern Australia, some areas of the Caribbean, and Mexico. It's the causative agent of melioidosis, and infection occurs via exposure to contaminated soil and water. Symptoms of melioidosis can vary greatly, so it can often be misdiagnosed as other illnesses. It's also important to note that melioidosis can be chronic, as well as it can reactivate itself years after initial infection. For this reason, it's also sometimes termed the Vietnamese time bomb because of its observance in many soldiers years after returning home from the Vietnam War. Interestingly, we have seen several cases of B. pseudomallei in recent years in New York State. These cases have been associated both with travel and immunocompromised individuals. There have also been cases of B. pseudomallei isolation in surrounding states such as Massachusetts as well. On blood agar, B. mallei may show up as pinpoint colonies after 24 hours, but the best growth is observed after 48 hours of incubation. Colonies at this point appear smooth, non-hemolytic, gray, and translucent. McConkey plates either yield no growth or pinpoint growth after 72 hours of incubation. On Mueller-Hinton agar, the colonies do not exhibit pigment, and B. mallei will not grow at 42 degrees. B. pseudomallei grows a bit more quickly than B. mallei, with growth of smooth, creamy, white colonies appearing on blood agar after 24 hours of incubation. After 48 hours, colonies tend to develop a metallic sheen and then turn more dry and wrinkled the longer that they are incubated, similar to Pseudomonas diphtheria. Pink colonies can be seen on McConkey after 1 to 2 days of incubation. However, it's important to note that the pink color is due to oxidation, not fermentation. Colonies are non-hemolytic and non-pigmented on blood agar and Mueller-Hinton agar. Unlike B. mallei, B. pseudomallei will grow at 42 degrees after 48 hours. B. mallei is said to have no distinct odor, while B. pseudomallei is said to have a musty, earthy odor. However, the practice of plate sniffing is strongly discouraged in the clinical laboratories due to increased risk of infection. While both organisms are Burkholderia species, they look very different on blood agar and chocolate agar, as can be seen in the photos. Colonies of B. pseudomallei are small, smooth, and creamy after 24 to 48 hours, where colonies of B. mallei are pinpoint to small gray at 24 hours and gradually become smooth gray and translucent at 48. On the gram stain, B. mallei appears as faintly staining gram-negative coccobacilli in pairs, bundles, and/or palisades. Some cells may also appear small, straight, or slightly curved with rounded ends. Gram stain of B. pseudomallei will yield small gram-negative rods that will either be straight or slightly curved. They usually form irregular arrangements of long, thin bundles. Like in the case of Yersinia pestis, bipolar staining can be observed in direct specimens and peripheral staining in older cultures, which can mimic endospores. Biochemically, unlike other Burkholderia, both B. mallei and pseudomallei are arginine positive. Additionally, B. mallei and pseudomallei are catalase positive and indole negative. B. mallei will be non-modal and oxidase variable, while B. pseudomallei will be modal and oxidase positive. Some other helpful tests for the identification and differentiation of these two organisms are their susceptibility to drugs such as polymyxin B, colistin, penicillin, and augmentin, as well as their growth at 42 degrees. While the biochemicals and morphology characteristics for Burkholderia and the other organisms we have discussed in this presentation are helpful in their identification, please feel free to reach out to the Biodefense Laboratory should you encounter some biochemicals or characteristics that don't match the typical criteria. For example, last year we were notified of a potential B. pseudomallei isolated from a canine that was adopted from Thailand, but this organism ended up being non-modal. Additional characterization, including whole genome sequencing, was performed to confirm that this was indeed B. pseudomallei. This is a perfect example as to why notification to an LRN laboratory is so important. In many cases, B. mallei and pseudomallei can be differentiated from other organisms based on lactose fermentation or non-fermentation on McConkey. Burkholderia cepacia can be differentiated based on its resistance to amoxicillin clavulanic acid, and colony morphology can help in the differentiation between other organisms such as Pseudomonas aeruginosa and Chromobacterium violaceum. Lastly, we will discuss Brucella. 3 specific Brucella species fall into the select agent category. Brucella abortus, suis, and melitensis. Brucella is zoonotic and is the causative agent of human brucellosis infection. Brucellosis is most commonly acquired through consumption of unpasteurized dairy products such as milk and cheese, but it can also be acquired via exposure to aerosols of the organism. This, in conjunction with its extremely low infectious dose, contribute to it being one of the most common laboratory-acquired bacterial infections. In New York State, we have several brucellosis cases each year, some of which we will discuss later on in the presentation. Brucella is characteristically slow-growing. A haze or dust-like growth can be observed on blood agar and chocolate agar after 24 hours of incubation, but at least 2 days of incubation is needed to see colony morphology. At 48 hours, the colonies will be small, white, and non-hemolytic. Carbon dioxide may be required by some strains. In the case of blood culture, Brucella grows slowly in aerobic bottles, usually needing at least 2 to 4 days, and it usually only grows on subculture after 48 hours. Brucella typically does not grow in McConkey, although some literature indicates that pinpoint colonies can be observed after longer incubation times. Brucella on the gram stain is extremely tiny and can sometimes be missed if a smear contains a lot of background material. It's a faintly staining gram-negative coccobacilli, and it usually arranges in singles which are not clustered. Brucella has a tendency to retain crystal violet, especially in blood culture smears, so under decolorizing can cause it to appear as a gram-positive cocci. Biochemically, Brucella is catalase, oxidase, and urease positive. It should be noted that oxidase may be variable, and the test should be performed on fresh cultures that are only 18 to 24 hours old. It's non-modal, and it's also negative in the satellite test. These are some more routinely isolated organisms in the clinical laboratory that Brucella can commonly be misidentified as. As you can see, with the exception of some Haemophilus species, all of these organisms are catalase, urease, and oxidase positive, just like Brucella. Growth on McConkey and tests such as motility and satelliting can be helpful in differentiating these organisms from Brucella species, as well as the source the organisms are isolated from. For example, Oligella is traditionally a urinary pathogen, and that, along with Bordetella bronchoseptica, are rarely isolated from sterile sites like Brucella would be. Just some key points to remember. Immediately move work to a biosafety cabinet anytime a slow-growing gram-negative organism is isolated, especially if there is no growth on McConkey. Blood culture bottles should always be vented and subcultured in a biosafety cabinet. And please contact the Biodefense Laboratory anytime an organism is isolated that could potentially be a biothreat agent. If you found the information presented today useful, please feel free to contact us for a set of bench cards outlining this information.
Now we're going to focus on some clinical cases involving Brucella exposures that have happened in New York State, as well as the causes of these exposures. I will start by reviewing a little bit of background on MALDI-TOF MS, as many clinical laboratories are beginning to acquire this type of instrumentation. I will talk about some facility and safety considerations that go with implementing MALDI-TOF. Finally, I will discuss some recent clinical cases involving Brucella exposures within New York, including the Wadsworth Center. MALDI stands for Matrix-Assisted Laser Desorption Ionization Time-of-Flight Mass Spectrometry. It involves an ionization source which creates the ions in the sample as a result of pulsed laser irradiation. MALDI is a soft ionization technique that prevents decomposition of fragile biomolecules, producing primarily singly charged intact protein ions. These ions accelerate down a flight tube after a uniform electromagnetic force is applied. The ions get separated by their mass-to-charge ratio, causing lighter ions to travel faster and reach the detector before heavier ions. For one system, isolates can be identified following one of 3 different methods. The first method is considered the direct smear. The isolate is selected from an agar plate and transferred to a moldy target. The isolate is then covered with matrix, allowed to dry, and placed into the moldy. The moldy generates a spectrum which is compared to a database to determine the identification of the organism. An alternate method is the extended smear. This is similar to the direct smear, except after the isolate is transferred to the moldy target, it is overlaid with 70% formic acid. After the formic acid has dried, it is overlaid with matrix, dried, and placed into the moldy. The last method is the tube extraction. For this method, an isolate is extracted using formic acid, ethanol, and acetonitrile. After extracting, the solution is spotted on a moldy target, allowed to dry, and overlaid with matrix before being placed in the moldy to be analyzed. When using moldy for identifying microorganisms recovered from patient specimens, general safety considerations include the potential for chemical and biological exposures. Some of these considerations include examining or manipulating cultured microorganisms, handling prepared target slides before or after MALDI-TOF-MS analysis, safe handling of primary patient specimens to prevent laboratory-acquired infections, and direct contact with reagents used to prepare samples for analysis, including matrices and compounds used for microorganism inactivation or extraction, as well as the exposure to chemical fumes created during stock solution preparation, target slide cleaning procedures, or inactivation and extraction procedures. MALDI-TOF-MS for microbial identification uses a phenolic acid matrix that is solubilized with organic solvents such as acetonitrile and/or ethanol. Additional chemicals such as formic acid or trifluoroacetic acid may also be used in protein extraction procedures, inactivation protocols, or to clean reusable target slides. Generally, small aliquots of matrix and formic acid solutions can be handled safely on the benchtop during target slide preparation and on-target extraction. These procedures should include the use of gloves, protective clothing, and be performed in a well-ventilated room. Other processes may involve larger volume and require a higher level of containment, such as a chemical fume hood or a Class 1, Type B1 or B2 biosafety cabinet. There are a few biohazard considerations that need to be taken into account. The highest risk occurs during handling, manipulation, and disposal of primary specimens and cultured microorganisms before analysis. The direct transfer onto a moldy target can be performed safely on the open benchtop using BSL-2 practices and facilities when a face shield or barrier are used. Otherwise, a biosafety cabinet is preferred. It is important to note that matrix alone might not totally inactivate microorganisms, as spots can differ based on the biomass thickness as well as if the entire spot gets covered with matrix. Every lab should adopt and verify the recommended inactivation protocols. Additional information regarding high-risk pathogen inactivation of microorganisms is available through a recently published journal article in ClinMicro. Despite enhanced biosafety education and improved lab practices, better engineering controls and biocontainment equipment, lab-acquired infections continue to pose a risk to personnel working in medical and veterinary laboratories. If a high-risk infectious agent is suspected, labs should consult with the appropriate reference lab first. Analysis of such isolates should be avoided, as culture manipulation may heighten the risk of environmental contamination and/or personnel exposure. In the event that an unsuspected high-risk or select agent is inadvertently analyzed on a MALDI-TOF-MS with or without an antecedent inactivation step, the incident should be assessed immediately and reported to the lab management in accordance with the lab's biosafety and infection control procedures. Risk management steps should include determining which lab workers were potentially exposed to the agent without appropriate safety measures, potential need for post-exposure prophylaxis and/or health monitoring, gathering any cultures containing the hazardous agent, label and sequester them, prepare contaminated disposables for off-site autoclaving, and thoroughly cleaning all of the infected bench areas. It is also recommended that if your MALDI instrument contains an air intake or exhaust filter, that this should be replaced as well. Some additional safety measures include knowing the limitations of your database. Each manufacturer has different organisms in their database, some of which do not include biothreat agents. A separate biothreat database would need to be purchased in some cases. While closely related organisms can be used as an indicator of a potential biothreat, such as Bacillus cereus for Bacillus anthracis, other biothreat agents will result in no identification, as is the case with Brucella species. This makes it all that more important to review information that is available about the specimen, such as patient travel history, growth rate, gram stain, and biochemical results. Now I'll discuss some clinical exposure cases that have occurred recently. As was mentioned in the earlier presentation, it's important to recognize that various high-risk pathogens have very low infectious doses, as is the case with Brucella and Francisella. In 2015, an alert was sent out by New York State and New York City making clinical laboratories aware of 4 incidences in 3 months of unexpected isolation of Brucella species, which required that steps be taken to address possible laboratory exposures. While many factors led to the exposures in these clinical laboratories, one common issue was that several exposures related to new instrumentation, including the MALDI-TOF MS. Something to note, we have not had any cases of seroconversion in the last 10 Brucella exposure cases. However, members of the Laboratory Response Network have worked with labs to provide information on prevention of lab-acquired exposures and has recently conducted a safety study focusing on MALDI-TOF for biosafety concerns. Some of the recommendations from the study include performing the tube extraction as the preferred isolate preparation method. We know that most labs do not want to perform this method, but it can greatly reduce exposure risks. Also, to reiterate, certain agents do not get fully inactivated on the target by matrix and/or formic acid. In 2016, 3 laboratories, 2 clinical and 1 reference laboratory, were exposed to Brucella from 1 patient. This patient had immigrated from Mexico in 2015, became ill, and visited Hospital 1. Hospital 1 identified the isolate as Haemophilus. The isolate was shipped to the Wadsworth Center for confirmation. However, RT-PCR ruled out Haemophilus. There was no indication of Brucella until the second isolate was received and identified. Simultaneously, while we were working on the first isolate, a second isolate from the same patient was being worked on as the same patient ended up going to a second hospital. Hospital 2 couldn't identify the isolate and submitted it to the Wadsworth Center as an unknown gram-negative coccobacillus. At the Wadsworth Center, the specimen was originally worked up in the biosafety cabinet. However, initial biochemicals did not rule in Brucella, so along with the isolate from Hospital 1, this isolate was prepared for MALDI-TOF MS, both of which on the benchtop. Our MALDI instrument does contain biothreat organisms, and the isolates were identified with high confidence as Brucella. The isolate was moved to our BSL-3 laboratory, where it was confirmed as Brucella melitensis. After this event and reviewing the rule-out algorithm, we realized that the current algorithm was overly complex and did not place enough emphasis on the gram stain results, and organism growth rates. The algorithm originally listed all the expected reactions for all select agents and did not specify the key tests that should have been highlighted. This was also an indicator that we should need to spend time retraining individuals on the algorithm. Why did all these exposures happen? Firstly, the rule out was overly complex and needed to be simplified. Additionally, aerosol-generating activities were carried out, which is why it is important to evaluate laboratory practices and reduce aerosol-generating procedures when possible, such as using disposable inoculating loops instead of sterilizing metal ones. Finally, moldy target was spotted on an open bench without the use of a facial barrier. While at first glance, the new algorithm seems overly complex, it is actually simplified. The change in the algorithm is to immediately send isolates to the biodefense lab that are slow-growing and have suspicious gram stains. Previously, isolates were sent to the biodefense lab only after performing biochemical tests. This gives us rapid detection of select agents such as Brucella. Additionally, you will notice that the use of biosafety cabinets throughout the testing procedure until select agents are ruled out. Most recently, in the beginning of 2017, a large clinical laboratory received a specimen from a patient with travel history from Mexico. After 3 days, the blood culture bottle flagged as being positive. The laboratory ran oxidase, urease, and catalase on the open bench, all of which were positive. After approximately 24 hours, small faint colonies were seen on the blood plate, but the laboratory decided to run it on the moldy tuff for identification. When setting up the moldy run, the target was spotted on the open bench without facial barrier. Not surprisingly, the moldy was unable to identify the organism. Additionally, a gram stain was run, which was read as a gram-negative coccobacilli. It is important to note that this laboratory is constructed as a large open room with many techs. This means in the event of an exposure, there's an increased risk of having exposed personnel and a decreased chance of determining where personnel are during aerosol-generating procedures, thus classifying the entire lab as high risk instead of no or low risk. And therefore, the entire lab would need to go on prophylaxis, which is a very hard course of rifampin and deoxycycline for 60 days. If an individual is immunocompromised, they would need alternative therapy or symptom watch, both of which are not ideal. Additionally, 24 weeks of serology is necessary. What could have prevented possible exposures? First and foremost, the information that is available needs to be considered before continuing with testing, especially focusing on gram stain results and growth rates. In this case, the organism was a slow-growing gram-negative organism. Second, if possible, which we understand is not always the case, it's advised that work be performed in a biosafety cabinet until it is deemed safe for the benchtop. Every laboratory should conduct a risk assessment and determine which laboratory procedures can cause aerosolization, directly increasing exposure risk. Some of these procedures include venting of blood culture bottles, which should always be vented in a biosafety cabinet, vortexing, and sonicating. Moldy should be a last resort and avoided when potential high-risk pathogens are suspected. It is also important to remind medical staff to alert the laboratorians when a patient has symptomology characteristics of brucellosis or has travel history in areas where Brucella is endemic. After so many subsequent Brucella exposures and alerts sent out by New York State and New York City, we were beginning to think that no one was reading them, but to our pleasant surprise, someone was listening. In 2017, 2 clinical laboratories received a specimen which ended up being positive for Brucella. The laboratories were the laboratories that I had previously talked about, which had multiple exposures back in 2016. Only this time, all work, which included culturing, subbing, and setting up gram stains were performed in the biosafety cabinet. No additional work, including MALDI, was conducted. Lo and behold, there were 0 exposures. Why were there no exposures this time? For starters, the biosafety cabinet was used for the unknown samples when the organism was determined to be slow growing. The blood culture bottles were vented in the biosafety cabinet along with setting up of the gram stain. The laboratories took it upon themselves to review ASM protocols for ruling out and referring potential BT agents after the initial exposures occurred in 2016. Along with contacting Wadsworth, they did not use any type of automated identification system. Some additional safety measures that the laboratories had implemented after the original exposures included benchtop shields and face protection. If you are able to acquire a MALDI-TOF or any type of automated identification system, there are some other considerations to think about. Laboratories will need to revise and update their risk assessments. You want to define the workflow and determine when chemical and biohazard exposures might potentially occur. You will have to develop protocols to mitigate these risks. Additionally, we are always very focused on the biohazards, but we cannot ignore other hazards such as the physical hazards like electrical shock, vibration, and noise. It is important to have knowledge of who's in the lab as far as non-laboratory personnel, as this has often complicated Brucella exposure investigations. This could include janitors, students, instrument service technicians that could all be potentially exposed if a highly infectious agent is isolated in the laboratory in their presence. Along with any new instrumentation, each lab should have a formal QA program that includes documented initial training and annual competency assessments. The competencies should include all specific tasks. For example, with MALDI, the competency must include everything from extracting and spotting the targets to running the instrument and cleaning the targets. A panel of well-characterized isolates should be used for training and competencies to ensure consistency among users. This should be a mixture of easily analyzed and more challenging isolates. More importantly, the training needs to include recognizing isolates that should not be analyzed by the MALDI-TOF MS. A simple checklist like this can be adopted for use in competency assessments for this method using the 7 criteria of evaluation. And with that, I would like to thank everybody who has listened to this presentation, and if you have any further questions, you may contact the Wadsworth Center Biodefense Laboratory.